Hello, and welcome to our section about population genetics. We're going to talk about some very interesting topics today. We're going to start with looking at the difference between microevolution and macroevolution and what that means. We're going to look at population genetics. What is that talking about? We'll look at the gene pools and what allele frequencies, what that means. We're going to look at a Hardy-Weinberg model. It's a mathematical model that shows us how we can think about population genetics. And then last, we are just going to look at what affects the gene pool. There's a lot of different things that affect that. First, just a review, evolution means a change in populations through time. We already learned that. There are two aspects to that change. And that's where we get the difference between the macroevolution and microevolution. When we're talking about a change over a very long period of time, we're talking about macroevolution. Very long period of time. It's usually the scale then for that is looking at the origin of new species. Microevolution, on the other hand, is talking about time within dozens or hundreds of generations. So that sounds like a long period of time, but that would be a much smaller period of time than what we're looking at in macroevolution. So microevolution deals with dozens or maybe hundreds of generations, and it's a change within a species that occurs. So macroevolution is talking about origin of species. Microevolution is talking about just within a species. We're looking at changes that occur. So that's the difference between those two types of evolution regarding time and scale. What do we mean then when we use the term population genetics? When we talk about population genetics, we're talking about a field of biology that studies microevolution. So we're looking at dozens to hundreds of generations, sort of smaller scale in the grand scheme of things, and we're looking at just changes within a species, not the origin of new species. So when we're looking at that, changes that occur within a species, here's just a silly little picture that helps you get the idea of what a gene pool looks like. We talk about all the genes of a local population of organisms. That's the gene pool. This is just a picture representing one particular Mendelian trait, and all the different alleles that go into that pool, that gene pool for those organisms in a local population. Now that you know what a gene pool is, we can look at another term, allele frequency. Don't let that scare you. Frequency, in biology, we're looking at proportions of percentages. How much out of the total do we have of a particular allele? It's the proportion of that allele among all the allele copies being considered in the entire gene pool. We can write it as a percent, or we can write it as a number. In this case, we're looking at 7. If we just look at the gene pool here, we've got all these different organisms contributing for this particular trait. There are different alleles to this pool. And if we look in this pool, we have a total of 14 alleles. 7 are capital B dominant, 7 are the recessive alleles. And if we look at that, allele frequency for the dominant allele, we're looking at 7 divided by 14. That gives us the number 0.5. So we can write it as 0.5. We can also write it, if we take the 0.5 and simply multiply it by 100, gives us 50%. Same thing. Means the same thing. The 0.5 means the same as 50%. The recessive allele also has the same frequency. The allele frequency for the recessive allele is also 50%. So let's look at 3 generations of some flowers here, and we're looking at the parental generation up here, and the first generation and the second generation of offsprings. And let's look at the frequency of the different alleles here. If we take this parental generation here, and we look at the allele frequency for the capital P, and the allele frequency for the lowercase p. Those are going to be hard to differentiate, but we'll try and do that. We have 2 dominant and 2 recessive out of 4. So 2 out of 4, that gives us 1/2. When we divide that out, it's a 0.5 or 50%. We get the same for the little p, 0.5 or 50%. Let's look at the F1 generation. Again, for our dominant alleles, we have 2 out of 4, and the recessive, we have 2 out of 4. So they're the same. We have the same allele frequencies. So here the allele frequencies are the same as for the parental generation. Now let's take a look at the F2 population. Let's look at the big P. How many of those do we have? 1, 2, 3, 4, 5, 6, 7, 8. 8 out of how many total? 2, 4, 6, 8, 10, 12, 14, 16. 8 out of 16. Once again, it's 0.5 or 50%. Well, the little p is going to be the same, 0.5 or 50% when we do the math. The frequency of each allele in each of these 3 generations, it remains the same. Now, I'll just give you a little bit of background. Because of this interesting relationship here that we have from generation to generation where the allele frequencies are staying the same, some mathematicians came up with a really great model called the Hardy-Weinberg Model, and there's some pictures of 2 of the men that they did not work together on this. They actually came up with it separately, and Hardy is the last name of one of the mathematicians and Weinberg the other. Another scientist also came up with this model, but he's just not included in the name. These 2 are the only ones that are included. And they came up with a mathematical formula, a model, that predicts the simple relationship between the allele frequencies in the gamete pool of a given population. It also predicts the relationship between the genotype frequencies. So remember the difference between allele frequencies. We've been talking about individual alleles. There are also genotype frequencies. We can look at the genotype if we look at what we were looking at before with our gene pool. A genotype here would be expressed as whatever that dominant feature is. This genotype, the phenotype would be here expressed as the recessive. Here's another genotype. But we can look at genotype frequencies as well as individual allelic frequencies. This model also is going to give us, it's very idealized. So it is a model for a polymorphic population, one that has 2 or more alleles of a gene for a trait. If there's only one allele present, obviously the model doesn't work, and there needs to be at least 2 different alleles or more. And the idealization of the model is limited, and we won't go into that so much here, but you can look into that for some extra. The Hardy-Weinberg model assumes some different things, that there is negligible mutation, the organisms are not mutating, that natural selection is not operating, or else we start getting a shift. The allelic frequencies will not remain the same if natural selection is in operation. It assumes that it's a pretty large population size. We're going to talk about that a little bit later. Population size, if it's really tiny, some strange things start to happen in the population. So the Hardy-Weinberg model is a very idealized model, and it happens only when some of these certain circumstances are met. Now let's take a look at how this model operates and what the 2 big mathematical formulas are. They are extremely simple. they take a little bit of work to work with, and there is another video that you can access to look at some advanced proficiencies and go ahead and work on how to put these formulas into practice and calculate different allelic and genotypic frequencies using them. What it assumes is P plus Q equals 1. And I know that sounds really strange. What is P? What is Q? And why is this equaling 1? This P represents the allele for purple flowers. It's the dominant allele. Whatever our dominant allele is, it's represented by P. Then we have a Q. Q is the allele for white flowers in this case. It's the recessive allele. And when you add the dominant allele plus the frequency of the dominant allele plus, we could write frequency here, plus the frequency of the recessive allele, we should equal 1. If you'll remember from our previous example, we had the frequency of the dominant allele is 0.5 and the frequency of the recessive allele at 0.5. 0.5 plus 0.5 is 1. So that works out really nicely, and it always does. The two added together will always equal 1. Now, by definition, we can use the definitions of what we just discovered and put it into this formula that always holds true when a population is in Hardy-Weinberg equilibrium and meets those ideals of the different Hardy-Weinberg rules. We have here P squared. P squared, as a term, represents the frequency of the homozygous dominant genotype. Then we have right here 2 times P times Q represents the frequency of the heterozygous genotype. And then right here, Q squared represents the frequency of the homozygous recessive genotype. So once we know the allelic frequencies of each of our, the frequencies of each of our alleles, we can plug those values into this equation and we can determine the frequency of the different genotypes. What is the proportion, what is the percentage of individuals that have that particular genotype? What is the percentage of individuals in a population that have that genotype? And how many individuals in a given population can we expect to have this genotype? And that's how we can use these formulas. So what is the significance of this Hardy-Weinberg mathematical model? Right here, just for a little aside, here are some Hardy-Weinberg assumptions. A question you could ask yourself: why would these things be important to maintain that Hardy-Weinberg equilibrium, to make sure that there is not some drift in these allele frequencies? Why are these things important? These actually essentially never occur in nature, by the way. That's why Hardy-Weinberg is an ideal case, because these things don't happen in nature. In nature, we have all kinds of variations going on and breaking the Hardy-Weinberg rules all the time. But the significance of the model is that allele frequencies in a population can be used to calculate all of the genotype frequencies, as we just talked about. Also, the allele frequencies are stable over time, and genetic variation does not disappear. If a gene pool is polymorphic, it will stay polymorphic. What does that mean? Remember, a polymorphic population has at least 2 different alleles for that trait. So that recessive allele does not disappear. Genetic variation does not disappear. The recessive allele is maintained because of the heterozygote genotype that is in the population. And you don't ever see in this Hardy-Weinberg model, you can go generation after generation and continue to calculate the different allelic frequencies and genotype frequencies, and you will continue to get a polymorphic population. That recessive allele does not disappear. It's hidden in that heterozygote genotype. One other really important thing to mention, Darwin was unaware of this model. He came up with his ideas prior to the Hardy-Weinberg model being developed. He was also unaware of Mendel's laws of inheritance. So when he was writing his theories and his ideas, he did not have the benefits of these two different very powerful ideas. He would have been really excited if he had, because some of the ways he tried to explain it were not very accurate. They didn't work out so well. So in a lot of ways, this Hardy-Weinberg model helps to explain some of what Darwin was trying to predict. It's a way of showing that this genetic variation does not disappear. It is maintained, and we can look at how populations can evolve over a long period of time and how they can change. So, if that variation remains constant within a gene pool, how can a population change over time? What can cause it to change? And in essence, it's those different criteria. The key is that we just talked about to the Hardy-Weinberg equilibrium, those different qualifications for it to be an ideal population. Well, those are the things that make the population change over time. We have natural selection, genetic drift, gene flow, and mutation. Those are the main mechanisms that affect the gene pools. And so let's take a look at each one of those. We've already looked at natural selection. We had a whole section on that. You already understand that. I just really like this picture of natural selection. This still works. These guys have an outlet right there floating in the water. Maybe they will not be around to pass their genes on to the next generation. Anyway, just to review, natural selection is just that the increased reproductive success leads to increased allele frequencies. Certain alleles are selected in favor of, and some may be selected against. Poor reproductive success leads to decreased allele frequency. So these individuals maybe are going to have poor reproductive success and will have a decrease in their allele frequencies in the gene pool and in the population at large. 2, genetic drift. So that's something we haven't talked about yet. What is genetic drift? Genetic drift is simply random changes in small populations. We're going to look at some different kinds of genetic drift. When we have small populations, random changes can greatly affect the gene pool. There's something called the founder effect. That's one way that random changes can dramatically affect a small population. That is where we have a few individuals from a population. Here's our population of ladybugs. A few individuals, they go and start a new population. And this new population has a completely different allele frequency than the original. That's called the founder effect, kind of like they're founding their own little island here, and they have a new allelic frequency than their original population does. There's something called the bottleneck effect. It is also a type of genetic drift where a number of organisms is drastically reduced for a few generations, causing a reduction in variation in the gene pool. Here we have our organisms, and for some reason, maybe it's a catastrophe, some kind of a geologic event, or any number of things, flood, fire, climate change, something that causes the population to be greatly reduced. And in that case, then there would be a much smaller gene pool. and there would be less variation in it. We could end up getting, as you can see, if the population is reduced to the point where only this genotype is represented, you're ending up then down here with a greatly reduced genetic variation in the gene pool. So that's called the bottleneck effect. That can happen when there's a dramatic reduction in the number of organisms, and you get a reduced variation in the gene pool. So something to keep in mind is that genetic drift is definitely something right here that it does not have a huge effect on large populations, but if we have really small populations, genetic drift causes a loss of the genetic variation in the overall gene pool because it reduces the frequency of the heterozygote genotypes. And over a period of time, that reduction in the heterozygous genotype frequency leads to a huge change, huge loss of genetic variation. Those heterozygotes, remember, that's sort of where we maintain the genetic variation. The recessive alleles can hide in there and be passed on, and they can be maintained in the population, but that changes when you're decreasing the frequency of the heterozygotes. Cheetahs are an example of this problem because their numbers are so small. We actually see a huge amount of inbreeding, and you've heard that word, right? Inbreeding. Well, cheetahs are very inbred. Extremely small populations eventually become very inbred, and unfortunately the cheetah populations are so small that there is a huge inbreeding problem there. Something you can look up is a term called Inbreeding Depression, where fertility and survival of organisms in a population are reduced compared with populations that are not inbred, and you start seeing problems and really negative, bad kinds of traits that start happening, and it is not good and healthy for a population to be inbred. All right, let's look at another mechanism that affects gene pools, and that is gene flow. What's gene flow? A little bit different than genetic drift. Gene flow has to do with change in the gene pool resulting from migration of individuals between populations. So if we look at this picture, kind of represents that a little bit. Here we have population 1. We have some gene flow. They're moving into this population 2. And here's our geographic barrier. Here, this is shown with some deer. We have our western deer population, our eastern deer population. They're a little bit different. Different species there. We have some migration from this gene pool into this gene pool. We might have some migration back this direction. That is gene flow, where actually genes are being taken and uprooted from one population and put in another population. Another mechanism that affects gene pools, mutation, definitely affects gene pools. Mutation is just the direct conversion of one allele into another. Mutations usually arise at a very low rate. They're spontaneous. They're not predictable. They are usually due to a DNA replication error. And they usually are deleterious, or they can be neutral. There are not that many examples of beneficial mutations. There are some, such as mutant alleles that provide resistance to insecticides. Those kind of mutations will spread very quickly through a population of houseflies. I think that's probably pretty obvious why that would happen. That would be very beneficial to those houseflies. So here's some advanced ideas for you if you like math problems and you're good at math, and I expect a lot of you to do this because a lot of you are really good at math. I have listed right here a website. It's also on the Moodle classroom website. Go and watch more of a step-by-step example of how to work through some of those Hardy-Weinberg equilibrium problems. and do some of those problems. They're kind of fun to do. So I expect some of you to do that for advanced proficiency. Look up more about inbreeding depression, give examples. Research some scientists and mathematicians who founded population genetics. Also, how could two individuals have slightly different genes but produce identical proteins? Think back to some of the things we've talked about in previous sections. How could this allow for hidden genetic variation? So those are some advanced ideas for you all to do, and I look forward to seeing you in class.